Hi everyone, welcome back to my channel, and today we are going to learn about Tca cycle. Tca cycle have three different names. First one is citric acid cycle, second is Krebs cycle because Hans Adolf Krebs has proposed this cycle, and third is tricarboxylic acid cycle because citrate, cis-aconitate and isocitrate, all three are participants of Tca cycle, so it can be called as tricarboxylic acid cycle. The citric acid cycle is the most important metabolic pathway for the energy supply to the body because 65 to 70% of this ATP is synthesized in a Krebs cycle. And all these enzymes that are needed for the Tca cycle are located in mitochondrial matrix. Are located in this mitochondrial matrix. So first of all, let's know an overview of this Tca cycle. The acetyl CoA is a 2-carbon structure, and when it combines with oxaloacetate, which is a 4-carbon structure, it will give rise to the citrate, which is the 6-carbon structure. And while in the formation of alpha-ketoglutarate, it liberates a molecule of carbon dioxide. So this alpha-ketoglutarate is a 5-carbon structure. Now this alpha-ketoglutarate will be converted into succinyl-CoA, and in this process again a molecule of carbon dioxide is released. So this succinyl-CoA is a 4-carbon structure, and it will go and form the oxaloacetate. So it is an overview, the basic overview of this Tca cycle. Now let's learn Tca cycle step by step. First of all, pyruvate is converted into acetyl CoA via the enzyme pyruvate dehydrogenase, and here a molecule of NAD is converted into a molecule of NADH. We have already discussed acetyl CoA is a 2-carbon structure, so we can also say this is a connecting link. This step is a connecting link between glycolysis and Tca cycle. Now what happens is, when this oxaloacetate will combine with this acetyl CoA, it will form citrate by the enzyme citrate synthase. Now this citrate is a 6-carbon structure. This citrate is a reversible step, and this citrate is converted into cis-aconitate, and this is converted into isocitrate by the enzyme aconitase. This enzyme acts for both this step. Now this is a very important step that we have to know is isocitrate is converted into alpha-ketoglutarate. This is the irreversible step by the enzyme isocitrate dehydrogenase. And in this step, one molecule of NAD will convert it into NAD++ H ion. What we have already discussed is here a molecule of carbon dioxide is also released to make this alpha-ketoglutarate a 5-carbon structure. Now this alpha-ketoglutarate is converted to succinyl-CoA, which is a 4-carbon structure because here also a molecule of carbon dioxide is liberated. In this step also NAD plus is converted into NADH plus H ion by the enzyme alpha-ketoglutarate dehydrogenase. In this step, thymine is also act as a cofactor. So thymine also act as a cofactor. Now this succinyl CoA will convert into succinate by the enzyme succinate thiokinase. And here a molecule of GDP will convert into GTP. Now this succinate will convert into fumarate by the enzyme succinate dehydrogenase. And in this step, FAD plus is converted into FAD, and here riboflavin act as the cofactor for this step. Now this fumarate is converted into malate by the enzyme fumarase, and this is the reversible step. This is also the reversible step, and this is also the reversible step. This both are the irreversible step. Now this malate is converted into oxaloacetate through this enzyme malate dehydrogenase. And here also NAD plus is converted into NADH plus, H plus iron. So this is the TCA cycle, and by looking at this cycle we can think it is as a closed cycle, but it isn't, because TCA cycle is an open cycle. Many compounds enter the cycle and leave. Each intermediate of this cycle is connecting to the another pathway, and we will know how this is connected to other pathway. So this is an summarization table of TCA cycle. We have discussed it about already. Now let's see how these intermediates are connected to the other pathway as well. So here this citrate is also used for the fatty acid synthesis. Likewise, alpha ketoglutarate is used in the process of transamination. This succinyl-CoA is used in the process of heme synthesis. Likewise, oxaloacetate is used in the formation of glucose and in the process of transamination. Another very important thing that we have to know is this pyruvate can be directly converted into oxaloacetate by this enzyme pyruvate carboxylase. And this pyruvate can also be converted into malate by the enzyme malate dehydrogenase. Now let's learn about this role of the vitamins in this TCA cycle. So four vitamins play a role in this TCA cycle. The first one is thiamine. Second is riboflavin, third is niacin, and fourth is pantothenic acid. Okay, we have already discussed this. Thiamine is used as the cofactor for alpha ketoglutarate dehydrogenase enzyme, and likewise riboflavin is used as the cofactor for succinate dehydrogenase. And this niacin is used as NAD plus as electron acceptor for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. And this pantothenic acid is used as coenzyme A, which is attached to active carboxylic acid residues, that is the acetyl-CoA and succinyl-CoA. Now let's go and learn about these regulators of citric acid cycle. So here we will be reading about these inhibitors and what is very important to regulate this cycle. So first of all, when there is high amount of NADH and ATP, it will inhibit the enzyme pyruvate dehydrogenase. Likewise, increase in amount of ATP and citrate will inhibit the enzyme citrate synthase. Increase in amount of ATP and NADH will inhibit isocitrate dehydrogenase enzyme. Likewise, increase in NADH plus itself in succinyl-CoA will inhibit the enzyme alpha ketoglutarate dehydrogenase. These are the inhibitors of the citric acid cycle. Another very important thing is the availability of ADP. So it is very important for the citric acid cycle to proceed, because if there is low level of ADP, then the oxidation of NADH and FADH2 through the electron transport chain stops. So what happens is this NADH2 will increase, and we have already discussed how this NADH is going to inhibit all our enzymes. So there must be the sufficient levels of this ADP to regulate this TCA cycle. So it is very important. So the regulators of this TCA cycle are ADP, NADH, citrate, ATP and ADP. So this is all about TCA cycle, and it may be very difficult to memorize all these things, so you can read it again and again, and you can make a chart so that there will be the visual memory. So thank you for today, and hope you guys enjoyed. We'll meet up in next video.